Hello, everybody. My name is EricGarcia. I'm a postdoc at the Texas A&M University Corpus Christi, and I'm pretty excited to share our project with Dalalas Luta in collaboration with the Division of Fish and Wildlife and the Wildlife Sport Fish Restoration. We are using environmental DNA or eDNA to determine the distribution and the biological associations of the endangered rhoda blue Dancerfly or Isnura luta, Dalalas luta. So all the materials you're going to see in this presentation were worked by all these people. So thank you for all of these guys, Becca, Daisha, Bronson, Kevin, and myself. So first, I want to give you a quick introduction to Isnura, the genus. So it's part of the order Odonata, which includes dragonflies and dancerflies. So within that, the suborder Psychoptera is just for Dulalas for dancerflies. The family is Cynagrionidae and the genus Isnura. And so the genus has a lot of species, about 75 species that are all over the world. So very successful genus. And a lot of these species, they look very similar, but there are diverse male genitalia parts, so you can tell the species apart. In Luta, this is not a problem because we only have two species of Isnura, Aurora, and Luta. Aurora has a green thorax with an orange abdomen, and Luta is slightly bigger and it's blue. So it's very easy to tell them apart. Once you see them, both of them, it's actually very easy to tell them apart. So the rhoda blue dancerfly is endemic to Luta. It can only be found in Luta. And here you see the male and the female. The male is slightly bigger. The female has the same coloration, but the blue is a bit more pale. And it was only discovered in 1996 and described only in the 2000. And it ever since has only been observed in 3 streams in the Thalassa region in Luta. So as you can imagine, we don't know much about its biology. We don't know the distribution. The nights or the aquatic library has never been described, and we don't know any ecological associations about this species. So because of this, the species was listed as endangered species in 2015, excuse me. And since then, there has been several surveys in the island trying to look for the fly, but however, they haven't found new sightings. So we are unable to study this fly using just visual surveys, so we need to employ different strategies to study this animal. So this is where this project comes in, and the first goal of the project is to test the utility of eDNA to determine the fly's Dalalas distribution, and also to try to find out the community composition where we can find the fly. In addition, we're going to be describing the ecological associations and habitat associations that this fly might have. The final goal of the project is to collect inverts from the water to build a reference database of DNA barcodes of local species that we'll use in our own eDNA study, but could be also used in later future studies as well. So first of all, what is environmental eDNA? So eDNA refers to any DNA that is shed by a living organism into the environment. And so then the scientists could then collect samples from the environment. And so this could be sediments, it could be water, it could be animal products. The DNA, the eDNA can then be extracted, duplicated, sequenced, and once you have the sequences, those could be assigned to specific species. Once you have a species, then you can start building a community based on your samples type, or if you have different samples from different locations or different times as well. So in all this process, assigning the sequences to a species, they requires a reference database. So what is this reference database? So these are going to be your DNA barcoding sequences. So they work just like a regular barcode in the supermarket, where you scan it, it's going to tell you what the product is. So this sequence is going to be unique to an organism, to a species. So this way you can study endemic, invasive, and many other type of studies as well. However, what happens if you don't have the sequence, if the sequence is not known? This is one of the challenges of the eDNA studies. If you don't have a reference, then one way to go around the problem is you can build the reference yourself. So we're going to be doing that. Another challenge with the eDNA studies is to know which location in the genome you use as a barcode. So not every single location, not every single sequence can be used as a barcode. And so you need to choose which sequence to use depending on the type of organisms you're going to be studying. So it's a different challenge of the eDNA studies that we'll have to deal with as well. Okay, so first of all, we want to know where the Dalalas is in Luta. So as you can imagine, there was no eDNA, there's no DNA sequence, barcode sequence for Luta before this study. So we're going to be generating that. And to do that, we collected 10 individuals, 10 adults, and we removed a single middle leg from the animal. And as you say, the animals were handled by trained professionals. And we did observe, we follow the flies after removing the leg, and we observed that they were able to fly, and they survived and were able to do their normal activities as well. So once we have this sequence, we just need to get the environmental samples. And luckily to us, we know the dancerflies or the lalas, they use the water to reproduce, and the naiad or the larvae lives in the water. So their DNA is going to be in the water if the fly is there. So we're going to be collecting samples across all the water available in Luta and see if we can get a DNA match. So that's going to tell us where it is. Now we're going to, we're also trying to know why is it there. So we're going to be studying the ecological association. So every time we go collect water, we're going to be describing the canopy cover, the vegetation type, and also the water characteristics, depth, the flow rate, the pH, the temperature, dissolved oxygen, turbidity, conductivity. So this is going to tell us the preferences of the fly. And at the same time, we're going to reconstruct the community composition using the DNA metabarcoding, and this is going to tell us about potential predators, prey, and competitors at the different sites. All right, so now to what we have done, actually. So in November 2023, we collected a total of 30 sites across Rota, and these were basically all the freshwater available in the island. So 5 of those came from the Rota Resort, and these are artificial ponds that were made for a golf course, and nobody knows if the fly is using this water as a resource or reproduction. So we went and collect water at these sites. The other 25 came from the Tallahassee area. So this is a series of 8 streams, 3 of which the fly was observed before, but no one knows if the fly is using all of them as well. So we collected 25, and we tried to get a site from the top upstream or high elevation, middle elevation, and lower elevation for every single stream. However, you can see there's some area that we didn't have samples. That's because some of these sites were really hard to reach. So we had to carry the equipment in our bags and walk through thick vegetation, crawl under thick bushes, crawl over trees, go through a matrix of bamboo. And we had to hike pretty uneven terrain. So it took a long time to reach a lot of these sites. We had 3 weeks with us. So we were able to collect 30 sites. So once we get to each site, we describe the habitat with the visual censuses looking for the dalalas and also inverts in the water. And we collected water samples, 3 replicates representing microhabitats. So for example, still water versus running water. And we filter 1 to 2 liters of water to collect the eDNA in filters. They're called Starbucks filters. And we were pretty lucky to have a pump that had a system with a Pelican case with 3 pumps. So we could do 3 samples at the same time, the 3 replicates. So once we were done, the DNA was stored with this, put on ice, and we freeze the DNA once we reach the lab, and those were kept frozen all the way to Texas with a cold storage while traveling. All right, so whenever possible, we also collected inverts. And so we collected both the species of the lalas in the island, as well as aquatic snails, crab, crayfish, shrimp, spiders, cane toad, water skimmers, water flies, flatworm. And so we only collect a few individuals. That's all we need. We just need to get that barcode sequence. And we're going to be generating the sequences for this, and later can be added to other sequences of other organisms, other studies that they already have the sequences to build a comprehensive database of barcodes in Luta. So currently we're at the barcoding stage. And so that the DNA from the inverts has already been extracted, has already been sequenced, and we are going through those sequences right now. So we built a reference from GenBank sequences, and we're currently creating these CO1 sequences using Eugene and Finch TV, excuse me. So what is next? So next we had to process the eDNA samples that we already collected, but we also are gonna be recollecting the same 30 samples in this upcoming May. And this is to find out whether there's any seasonal variations in the ecology of the fly. So we invited you to stay tuned and look for our results perhaps next year around this time. Okay, some acknowledgements. The funding came from a competitive state wildlife grant. And there was a lot of people that helped us to do already in this project. So we have people helping us getting permits, collecting samples, analyzing data, and a lot of people gave us access, landlords that gave us access to the private land. So we could donate with all of these people. So this was massive to all of them. Finally, unfortunately, I couldn't be live, but if you have any questions, you can email me or you can scan this QR code, and I will take you to my website for more information. But I'm sure that Becca is going to be in the Zoom call, so if you have any questions, she could probably answer them for you. Thank you, everybody.